Dear students, as you know that we have already discussed Ti-Plasmids and the genes available on the Ti-Plasmids. And in the previous lecture, we have studied the Ti-Plasmid, and today we have also discussed the types of the Ti-Plasmid. And as you know that the Ti-Plasmids are of three three types, and these three types of the Ti-Plasmids are Nopaline Ti-Plasmid, Octopine Ti-Plasmid, and Agropine Ti-Plasmid. As you all know that Ti-Plasmid has four very important regions, and these four regions, as you know, include A region, B region, C region, and D region. And it is also known to you that this A region has a very specific part of the DNA, Ti-Plasmid DNA, which is known as tDNA region. And this tDNA region stands for the transfer DNA region. And this transfer DNA region has the genes, and this transfer DNA region remains differentiated or demarcated from the other part of the Ti-Plasmid by the right border region and left border region. You know, and this tDNA region has the very important genes, and these genes include auxin gene, cytokinin gene, and opine synthesis gene, and all such genes present on the tDNA region constitute oncogenes, which are responsible for causing and proliferating the tumor in the plants. In the plants, the tumor formation gene, these are responsible for the induction of the tumor and the proliferation of the tumor cells. So these are the genes are present on the tDNA region. And tDNA region is also very important because the genes, because if we have to use this Ti-Plasmid as a cloning vector, the desired gene is tagged at the tDNA in the tDNA region at the near the left border region and right border region. Therefore, this tDNA region of the Ti-Plasmid is very important. And in beside this, the region D has the genes for the virulence, which are known as virulence genes, and the number of virulence genes present on the Ti-Plasmid is 9. And if there is any gene mutation in the virulence gene, the capacity to cause the infection will be lost, and therefore these virulence genes are also very important, which is responsible for causing infection in the wounded part of the dicot plant. And the region B has the genes for the origin of replication, and in addition to it, the genes for the opine catabolism. And the region C has the genes for the transfer, and that gene is known as the TRA genes, and which is responsible for the conjugation. Now we have to discuss the different types of the Ti-Plasmid. Ti-Plasmid are of three types depending upon the genes present on them for the synthesis of a particular type of the opine. And on this basis, the Ti-Plasmids are of three types, and these three types of Ti-Plasmids include Nopaline Ti-Plasmid, Octopine Ti-Plasmid, and Agropine Ti-Plasmid. These three types of Ti-Plasmids differ from one another in some respect, which you have to write, which you have to know. So I have discussed the diagrams of all the three types of the Ti-Plasmid and the differences among them on the whiteboard. And I would like to show you the whiteboard. Look at the whiteboard and try to understand the differences among all these three types of Ti-Plasmids. Look at the whiteboard. Is the whiteboard visible, Sajia? Hello, Jubina? This is the
yes, sir.
The types of different types of Ti-Plasmids have been sketched on the whiteboard, and just I am going to explain at first the Nopaline Ti-Plasmid. This is a diagram of the Nopaline Ti-Plasmid. Look here, this is the Nopaline Ti-Plasmid. This is the tDNA region. Presence of a single tDNA region in Nopaline Ti-Plasmid, and the genes for Nopaline synthesis and is for genes for auxin, genes for cytokinin, and nopaline synthesis genes are located on the tDNA region. And right border region and left border regions are present also here, as you know. And left and right border region consist of 24 base. Okay. This D region has the barbarin genes. The C region has transfer gene. NOS, the gene for the nopaline catabolism is located on the B region, and the G and also the gene for origin of replication is available or located on the B region. Now look here, the main features of this nopaline Ti-Plasmid. Presence of only one tDNA region. But there is presence of only one tDNA region in nopaline Ti-Plasmid, and the length of the size of this tDNA region is 20 kbp. The presence of NOS and NOC genes, nopaline synthesis and nopaline catabolism genes, differentiation of tumor cells into the teratoma. ठीक है, tumor cells induced by this nopaline Ti-Plasmid proliferate more rapidly. ठीक है, look at the octopine Ti-Plasmid. This is the diagram of the octopine Ti-Plasmid. This is a very important feature of this octopine Ti-Plasmid is that there is presence of two tDNA region, right tDNA region and left tDNA region. So this is this is the right tDNA region, left tDNA region. And what does it show? It shows that in case of octopine Ti-Plasmid, there are two tDNA regions, whereas in case of nopaline and agropine, there is presence of only one tDNA region. In this Ti-Plasmid, presence of octopine synthesis gene and octopine catabolism genes are available. Origin replication of origin site is available in the B regions. Transfer gene is available. Violin genes are available. Look at the features. Presence of two DNA region, left tDNA and right tDNA. The size of the left tDNA region is 14 kbp, and the size of right tDNA region is 7 kbp. Presence of OCS and OCC, yani octopine synthesis and octopine catabolism genes. No formation of teratoma. Slow proliferation. The tumor cells induced by octopine Ti-Plasmid proliferates slowly. There is slow proliferation of the tumor cell. In case of nopaline, the tumor induced by nopaline Ti-Plasmid undergoes rapid proliferation. But in case of octopine Ti-Plasmid and tumor induced by it, the cells of tumor will undergo slow proliferation. Okay? In case of agropine Ti-Plasmid, look the diagram of the agropine. The TDS, the single tDNA region is present. The gene for agropine synthesis is available. Auxin, cytokinin genes are available. Agropine catabolism genes are available in the B region. Transfer gene are available in the C region, and violin gene present in the D and D region. So this is the agropine Ti-Plasmid. Let us see the feature. Presence of only one tDNA region. Size of the tDNA region varies from 15 to 18 kbp. Genes for agropine synthesis and agropine catabolism. No formation of teratoma, and slow proliferation of the tumor induced by the tumor cells induced by agropine Ti-Plasmid. And in all the three types of Ti-Plasmid, the other genes which are present here, auxin gene, cytokinin gene, okay, origin of replication, transfer gene, root inducing genes, these genes are identical. But the main differences among these three types of Ti-Plasmids are the types of the opine. In case of nopaline synthesis gene. In case of octopine Ti-Plasmid, octopine synthesis gene. In case of agropine synthesis genes. And beside this, the number of tDNA is also variable. In case of nopaline, one tDNA region. In case of octopine, two tDNA regions. In case of agropine, a single tDNA region. And the other difference is that the tumor cells induced by nopaline Ti-Plasmid undergo rapid proliferation, whereas the tumor induced by the octopine and agropine Ti-Plasmid undergo slow proliferation. In case of tumor induced by nopaline, there is no differentiation of tumor into the teratoma. There is differentiation of tumor cell into teratoma, but in case of octopine and nopaline, the tumor cell do not differentiate into the teratoma. So these are the differences among the nopaline, octopine, and agropine Ti-Plasmid. Am I clear to all of you? Am I clear to all of you?
Yes, sir.